One of the things that we have to do when we want to make spread plates or pour plates in order to determine an estimate of how many bacterial cells are present in a particular environment, one of the things we have to do is we have to dilute our sample. Now, I'm not talking here about recreational bodies of water or water treatment facilities or public water supply facilities, because of course those samples are generally already quite dilute. Remember, we often have to filter out those samples somehow in order to concentrate the cells that are present in the sample. Here I'm talking about other sample collection. Here I'm talking about samples that are collected from surfaces in places like hospitals and doctors' offices and care facilities and so on. Whenever we collect a sample and we attempt to determine how many cells are present in that sample, if we don't know at the start how concentrated the cells are, we could end up producing a plate that looks like this one up here on the left. This plate is covered in so many colonies that the colonies have actually merged together to just produce this one mass of cells. There's no way, of course, to count colonies here. There are just too many of them, and they've actually grown to the point where they're all touching each other and creating this mat. So whatever sample we used to make this plate, it had far too many cells in it for us to make a spread plate with directly. So to avoid this from happening, what we do with our samples when we get them in the lab is we create what's called a serial dilution. We're not going to make one spread plate from our sample. We're going to make a series of them, and each plate will be made with a slightly more dilute version of our sample. So we take our original sample and we dilute it typically by a factor of 1 to 10. Then we draw some of it out, put it on a spread plate or a pour plate if necessary, and put that plate in the incubator. We're not going to stop there though. We're then going to take that sample that was diluted 1 to 10, and we're going to dilute it 1 to 10 again, and again, and again, several times. And we're going to make a separate plate for each dilution. That's what you're looking at on this slide. This plate, which we can't even read, this plate was made with the original sample. This plate was made with the original sample diluted 1 to 10. There are still way too many colonies on this plate for us to count. This plate was made from our original sample diluted 1 to 10 a second time. In other words, this plate was made with a dilution of 1 to 100. 10 times 10 is 100. If we dilute the sample again 1 to 10, now we've made a plate that's 1 to 1,000. And then we can make one 1 to 10,000 and 1 to 100,000. You can see on this final plate there's only 2 colonies growing. When we diluted the original sample stepwise to 1 in 100,000, we diluted out the cells so much that there were only 2 cells that landed on this plate. 2 cells that grew into individual colonies. This plate isn't really much use to us either. It was simply too diluted at this point. So when we get a sample that has a number of cells in it that we just don't know, we're going to always do a serial dilution before we make our spread plate or our pour plate. We're going to sequentially dilute the original sample by a factor of 10. And we're going to do that several times. So we will plate our original sample. We'll plate the sample diluted 1 to 10. We'll plate it diluted 1 to 100. We'll plate it diluted 1 to 1,000, 1 to 10,000, 1 to 100,000. We can even go beyond 100,000 if we think we need to. But most times, about 6 individual plates is fine. What we're going to do then is we're going to grow all of these plates. We're going to allow the bacteria to grow in the incubator for about 24 hours. And then we're going to choose the plate that we're going to count. And the way we do that is we visually estimate which plate has between 30 and 300 colonies on it. Let me say that again, because that's an important number for us to remember. We're going to choose a plate that has between 30 and 300 colonies on it. That way we know we have enough colonies to get a good estimate, and a small enough number of colonies that we can actually count and count accurately. So in this particular example, this is the plate that we would use. Now, when you take this in for counting, you don't actually have to count each and every individual colony. More typically, what we do is we divide the plate into quarters, and we count the number of colonies in 1 quarter and then multiply it by 4. And that will give us an estimate of how many colonies are on this plate. Remember, we use that colony number to then estimate how many cells were in the original sample. When we're trying to determine how many microbes are present in an environment, we're faced with a lot of different kinds of samples that we have to evaluate. So we're often using different units of measure depending on the type of sample. For example, if we're trying to evaluate flat, hard surfaces in, let's say, a long-term care facility, we're often evaluating how many cells there are per surface area, perhaps centimeters squared. Or if we're dealing with a water sample, now we're trying to calculate cells per volume, for example, per ml. But regardless of what the original sample material was, by using serial dilution and the spread plate method, we can report back to whoever requested the sampling. We can report back to them how many CFUs, or colony forming units, were present per unit of the original sample. It might be 20 CFUs per centimeter squared, or 1,000 CFUs per ml. It doesn't matter. The important thing is that we are giving them an estimate of how many cells were in their original sample. Now remember, when you hear me use the word colony, what I'm talking about is something visible on the surface of an agar plate or an agar container. It's a visible mass of cells. There are so many cells in that little lump of material, we can see it with our naked eye. And each one of those arose from one cell. These are clones, in other words. They are genetically identical to each other. One cell landed on that spot on the agar, and after it incubated for 24 hours, that one cell divided and divided and divided and divided until it produced a little mound of cells, a colony, that we can see with our naked eye. Parent cells produce offspring or daughter cells through a process called binary fission in bacteria. We're going to be talking about binary fission next week. It may seem hard to believe that one parent cell can create thousands and thousands of offspring in a 24-hour period, but you have to remember that this produces an exponential increase in cell number. That parent cell divides and makes 2 cells, which then divide and make 4, and then 8, and then 16, and then we go to 32, and 64, and 128, and very quickly, through exponential growth, we end up with a little colony of cells, thousands and thousands of cells, all arising from that same parent cell, and all genetically identical to each other. We can count those colonies and report them as CFUs, which are an estimate of the number of cells in the original sample. Now, as I said a few minutes ago, if we're using a Rodak plate to gather our samples, there are actual published data tables that are available to us to let us know what our count actually means. What does it mean if I get 4 CFU on a plate that I used to sample, say, the floor in an operating room in a hospital? What does it mean if I got 8 CFU when I sampled a table that healthcare personnel use to eat their lunch in the hospital facility? These published tables give us the information we need to know whether or not that number that we generated is acceptable or not. Remember, a CFU is not the same as 1 cell. A colony-forming unit could have come from 2 or maybe 3 cells, so it's just an estimate. It's our best estimate of how many cells were in the original sample. In the laboratory, sometimes we're doing a lot of spread plates. It all depends on which laboratory you're working at and whether or not they do a lot of environmental sampling. You can imagine that in a lab where they only do a couple of spread plates per day, it's quite possible to simply count the colonies by hand. Like I said, you can draw quadrant lines on the bottom of the plate, then count the number of colonies in one quadrant and multiply by 4, and you'll get a good estimate of how many colonies are on the plate. If you're in a lab, though, that does a lot more spread plating, you're going to need a little bit of help getting your counts. You wouldn't want to sit and count colonies all day long. And there are tools available to us to make this job a little easier. Now, down here, you're looking at an image of a plate counter. This is a manual plate counter. The person will place their agar plate on top of this disc-shaped light source. You'll notice that there are lines etched into the plate here. When you place your agar plate on top of this, you can see those grid lines through the plate. And that makes it easier to count the number of colonies systematically, so you don't count the same colony twice. You'll also notice that the person's holding a stylus. This stylus allows the person to touch the surface of the agar dish where there's a colony. We're not touching the actual colony here, we're touching the bottom of the plate. And the stylus will keep track of how many colonies you've touched. That's what this readout is here. So the person takes that stylus and just touches the colonies. Touch, 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 touch. The person does not have to keep track of this number in their head because the machine's going to do it for them. Again, you count a quarter of the colonies, multiply it by 4, and you have your plate count. There are lots of different versions of these manual plate counters available. Again, they make it a little easier to do more plates a little bit quicker than just doing it 100% by hand. But sometimes these aren't even enough. We need a system that can do a lot more plates a lot more quickly. And of course, there's computer software for that. That's what you're looking at up here on the right. There's an agar plate that was placed underneath a camera that was associated with this software package. The camera shoots an image of the plate and feeds that image into the software. And what the software does then is count the colonies on the plate and report that back for you. You can imagine that this system is much, much faster, probably a lot more accurate, although you do have to buy the software for the laboratory to use. So we have manual ways to count colonies. We have what are called semi-automated ways. That's what this is. And we have computerized ways or fully automated ways to count colonies. It all depends on what your needs are as a laboratory and how many of these spread plates you're doing every day. Now, when it comes to the Rodak plates, I want to show what one of these published tables looks like, so you can see what you would actually be evaluating if this was your job in a healthcare facility. There are people as part of their job, are doing this routine environmental monitoring. Maybe once a month, they're going around and they're sampling surfaces in and around the hospital in order to test to see whether or not their disinfection protocols are working well. On this slide, you can see a range of values for colonies growing per Rodak plate. In other words, in the hospital setting, after surfaces have been disinfected, someone goes around and presses the agar in the Rodak plate against the surface and sends it to the lab to see what grows, or hopefully what doesn't grow. In this particular example that I'm showing you, if between 0 and 25 colonies grew, that would be considered a good result. But if between 26 and 50 colonies grew, that would be only considered fair. And if over 50 colonies grew on that plate, that would be considered poor. Now, individual institutions often hold themselves to stricter guidelines than these published guidelines that you can find in textbooks and so on. Institutions often hold themselves to a stricter standard, in other words. So if in the published table you see numbers like this, oftentimes what the hospital will say is, no, we're not going to call it good unless there's between 0 and 10 colonies on the plate. Here's one of those published tables that you find for these Rodak plates. You can see that across the top of the table we have on the left a column representing the areas that got sampled. And then over to the right we can see the number of CFUs that would be a good result, the number that would be a fair result, and the number that would be a poor result. Let's take a look at this. Under the sampling, we have floors listed, and then we have both the operating rooms and the OB, the obstetrical wards, and the nursery, so where newborn babies are going to be kept. This, you can imagine, these are places where we have to be very strict with disinfection in order to prevent hospital-acquired bacterial infections in the patients. So we're looking for between 0 and 5 CFU per Rodak plate. That we will consider a good result. If there are between 6 and 15 colonies, we're going to call it fair. If there's more than 16 or 16 and above, we're going to call it poor. Now, if the result came back poor, what's going to happen is the people in the hospital who are in charge of this, who are in charge of environmental safety, are going to want to examine what kind of disinfection is going on in the operating room and the obstetrical floor and the nursery, and they're going to want to change it and make it better, and then retest to try to show that yes, they're doing a better job eliminating bacteria from surfaces. Here's another example, tabletops. They're talking about tabletops probably generally throughout the hospital facility. We've got patient rooms listed, both the floor in the patient room and table surfaces in the patient room. You can see that the floor is a little bit of a looser standard. Anywhere between 0 and 25 colonies growing on the plate, we're going to call a good result. But if there are even 6 colonies growing on a sample taken from, let's say, the table that the patient eats off, we're going to call that fair. So we're going to want the tables that we serve food on to be at a higher standard of disinfection than the floor in the patient room. We've got bathroom surfaces, we've got other floors, we've got other surfaces. You can find these tables for all kinds of surfaces that get sampled in healthcare settings. Let's take a moment now and summarize the things we've been talking about so far today. In microbiology, we're often tasked with evaluating environmental samples for the presence of bacteria and other microbes that might be harmful to human health. If we have a liquid sample, we can quite accurately count the colony-forming units per unit of original sample using spread plates and CFU counts. If we're not working with a liquid sample, but instead we are taking samples from solid surfaces, we can do that using a sterile swab. Or we can use commercially available Rodak plates. With either of those methods, we're also going to be producing spread plates, and we're going to count CFUs. But this time, it's more about how well we're disinfecting that surface. We're not trying to identify what microbes are growing on a Rodak plate. We are using the Rodak plate to tell us how well we're getting rid of microbes in the environment. Now, there's one other thing for us to discuss about Rodak plate testing, and that's the limitation of it. Remember, we take these Rodak plates into locations like healthcare settings, long-term care settings, and so on, and we press them down onto surfaces. You can imagine that you cannot take a single Rodak plate into an operating room and get a good idea of whether or not that operating room is effectively disinfected. You have to take several Rodak plates. You have to press those plates against several different locations in the operating room in order to get a representative idea of how well that operating room is being disinfected. If I took one Rodak plate and I pressed it down onto, let's say, the surface of a desk, I'm only seeing if there are cells present right there in that spot. So it's very important that I don't take one sample, but that I take several samples across the desk so I can get a representation of the entire desk. We're going to switch gears now and leave environmental sampling behind so we can talk a little bit more about culture media. Remember, in the microbiology lab, we are often trying to grow microbes purposefully in pure cultures. Remember, a culture is just a collection of cells of a particular microbe that are growing in a container. In order to encourage those cells to grow and divide, we have to give them the things they need in order to divide. We have to supply basic nutrients in the culture media. The most basic things that bacteria need to grow are a source of water, a source of energy, in other words, carbohydrate, a source of carbon in order to build other molecules, and that comes in the form of protein and partially digested proteins called peptones, and finally a range of micronutrients, things like nitrogen, phosphorus, sulfur, and so on. If we supply these basic things to the bacteria, most of the time it will be able to grow for us, and we can create a pure culture within about 24 hours. It's probably no surprise to you now that we have lots and lots of different kinds of culture media available to us in the microbiology lab, and that's because we are trying to grow all different kinds of microbes. Some microbes have very basic nutritional requirements, but some of them have very complex nutritional requirements. And so we need to have appropriate culture media for each particular kind of bacteria that we're growing. We define culture media according to its chemical properties, and we're going to learn about 6 different types of culture media. Now, the first kind of culture media is referred to as chemically defined medium. Like the name suggests, this is a culture medium that we know every ingredient in. In other words, it has a very simple recipe of known ingredients. In comparison, there's a second kind of media called a complex medium. What a complex medium has in it is some kind of an ingredient that comes from nature, but that has a composition that is not 100% known. For example, sometimes we add animal blood to culture media, or eggs. Sometimes we add different types of milk or meat extracts. These products provide the media with very specific types of nutrients that individual microbes might require. But because they are complex natural ingredients, we don't know exactly everything that's inside of them. A chemically defined media, in comparison, is one that we know exactly what's in it. Chemically defined media are typically used to grow what we generally refer to as non-fastidious bacteria. Complex media are used to grow fastidious bacteria. All that word fastidious means is difficult to grow, or microbes that have very particular, very specific and exacting nutritional requirements, and so they're harder to grow. There are several general purpose, chemically defined media available to us commercially. We buy these media in a powdered form, and we constitute the media in the laboratory so we can use it. The most common chemically defined media come in both a solid form or an agar form and a liquid or a broth. One of the ones that we use quite commonly is just referred to as nutrient media. It's a chemically defined media, so it's good for growing organisms that are easy to culture, non-fastidious. Nutrient broth is available and nutrient agar is available. Now, I want to point out here that when I use the word agar, I'm referring to a material that's very similar to what we know as gelatin. Agar is a carbohydrate, and it's extracted from certain types of algae that grow in the sea. It's part of their cell wall. Agar is the matrix of our solid culture media. but we have to add in the nutrients to the agar in order to grow bacteria. The agar, in other words, is not nutritional. It's simply the matrix of that solid media. We have to add in the nutrients. Now, agar is a perfect material in the microbiology lab if you want to build a solid media, and here's why. You can liquefy agar at a temperature of about 100 degrees Celsius. Once it cools down, it stays solid, including in the incubator. We typically grow bacteria at a temperature between 35 and 37 degrees centigrade, which is warm. That's much warmer than room temperature. That's closer to body temperature. So we can liquefy at a high temp. That will allow us to pour the agar easily into, say, petri dishes or even test tubes. But that agar will solidify as the temperature comes down, and it will stay solid at the most common temperatures that we incubate at. Agar, when it's solid, produces a nice smooth surface, and it allows colonies to grow quite well. It's very easy to add our ingredients to liquid agar. And as I said, very few microbes get any kind of nutrition from agar, so they're not going to break it down. They're not going to degrade the agar. You can imagine that would be a problem. If the microbes could actually eat the agar, we wouldn't have nice colonies growing on the surface. Different types of culture media are readily available in a powdered form from different commercial supply houses. You can also purchase pre-made media, though. And sometimes when we need a very particular type of media for a very particular type of bacteria, it makes more sense to simply purchase pre-made agar plates. But if we're making lots and lots of a common media that we use every day in the microbiology lab, it makes more sense financially for us to make the media ourselves. We buy the commercial powder and we simply add in whatever nutrients we need to add in, and we make our own agar plates right in the laboratory.